Hi everyone, we're going to be going over gel electrophoresis today. So let's look at a real lab scenario. You may have, for example, we have this insert of DNA, and we've put this in a vector that's about 1,000 base pairs. So our 150 base pair insert in this vector is about 1.15 kb. And now in the real world in our lab, we're going to be dealing with samples where you actually can't really see the DNA. A lot of the cases, it's actually going to be a clear volume. It's not going to be colored, so there won't be some sort of indication as to whether you successfully inserted into your vector or your plasmid of interest. So gel electrophoresis is a technique that can be used for separating out DNA and checking samples to see if you did a successful PCR or you did a successful cloning or anything really where you want to separate and check your DNA in any of your samples. So we'll take a little bit of the sample out and then we can run it through gel electrophoresis. So just for today, if I wanted to check my vector, I'm going to be doing a restriction endonuclease digestion. So I'll be cutting the DNA at these sites along the vector. I'll be cutting out the insert here and then linearizing the vector here. If we don't have the insert, we'll be cutting right about here, and then we'll just get the vector. So taking that into the context of our samples, let's say that we have, 4 samples that we did, and let's say they're these here, because they're probably going to be clear. These 4 samples, 1, 2, 3, 4, and we did a digestion for all of them. We digested using a restriction enzyme, and now we want to check, okay, did our digestion work? Did our cloning work? Do we have our insert inside our vector? So these questions will be answered using our gel electrophoresis. So gel electrophoresis actually is somewhat simple. It works off some very good principles. The first is that DNA is actually overall has a negative charge. That means if we place it in a solution, you can see here buffer solution, that's what this volume is in this large container. If we place it in a solution, it's going to move towards the positive electrode. So if we had sample wells and we put our DNA inside, the DNA will move towards the positive electrode. The next thing is that we place it inside a gel called agar. So agar is a gelatinous polymer. You may have actually eaten some of it when you have some certain candies. Sometimes it's a substitute used instead of gelatin. You also will see petri dishes are made using more low-grade type of agar as well. So agar is a polymer. It's going to be filled with long strands of molecules, and it forms this kind of 3-dimensional matrix whereby if we take DNA will be placed inside the agar and will have to pass through this matrix. Just like that. So it'd be moving towards the positive electrode. If we have a very long piece of DNA, something very long, it's going to take longer because it's going to have to pass through, because it's so large, it will have to find more ways to pass through. And it'll travel slower through the gel in the same amount of time that a, smaller piece of DNA may have passed through. So if we were to compare the two, this is what gel electrophoresis is doing. It's separating DNA based on its size. So let's kind of look at what's going on here. So here's an example of a casting, very simple, just we have our basically cutout rectangle and we have a flask here, and our flask contains our agar. Here we go, here's our agar. It's very hot, we just heat it up and we're gonna pour it inside. So just some things to know is that the agar contains a substance that binds to DNA. and it lights up in UV light. So what's going to happen is when we have our DNA inside of the agar, the DNA is actually going to be bound by molecules inside. So it can be different things. Sometimes there's ethylene bromide was commonly used a few decades ago. They're kind of phasing it out because it's a very potent mutagen. So it actually, binds between bases. So it's not very a safe chemical. So we have some alternatives. And then we put it under a camera and a light that shines UV, and all the areas with DNA will light up. So we'll be able to see where the DNA was. So we've waited for our agar to cool, and now it forms this block of, like this gelatinous material. And as it's cooling, so we haven't waited entirely for it to cool, I misspoke there, we're going to place a comb inside of the gel. So the comb is exactly what you'd expect, like with a hair comb, where it has these little notches that are going to rest inside, and it's kind of like putting a fork in Jell-O. So you're going to be placing a fork in Jell-O, you wait for it to cool, and then you take the fork out. You'll have little tiny holes where the fork was. So we'll place our comb inside, and then when we take it out, once the gel has cooled, you have these little holes that we call wells that go inside of the gel, and that is where we're going to place our sample. So if we have our pipette, we're going to pipette our sample inside, and the DNA will sink down to the bottom. Now when we put our DNA in, our DNA mixture will be combined with a dye. The dye will help us see where it's sunk inside the well. It's going to have glycerol, which is highly dense, especially compared to the buffer solution, so that it will sink down to the bottom. Otherwise, it might overflow outside, because this is all happening underneath a buffer solution. So this is all, I guess you could say, underwater in a way. So you'll be doing this. It's kind of hard to do, actually, sometimes when you do it for the first time. It can be hard to see where the wells are. So you have to add it carefully, and then what's going to happen is then we have our DNA in the sample, and those are going to combine with the agar so that we can see, when we have a short strand, it's going to travel very far in the same amount of time that a long strand might travel less far. It'll show up as a band on this gel, and that will show us, oh, hey, this is where it went. Usually our leftmost or our rightmost well will contain what's called a ladder or a marker. This is a solution with predetermined distances, so that you know that, okay, this band here will be 1,000 bases, this band here that will show up will be 500 bases, and then this one down here will be 50, for example. So here's kind of an example of what your gel might look like when you take an image of it. So here we have our empty wells on the top. We have our samples A, B, and C that we put inside the wells, and then we let the gel run towards the positive electrode, and all the DNA is slowly passing through. And you can see that the small pieces of DNA traveled the furthest, and when we compare them to our marker, our ladder, it matches up at approximately the 100 base pair distance. So we would conclude that this DNA fragment right here is 100 base pairs. So that's what we use. We can use gel electrophoresis to help separate them. So on the top right here, if we were to look at the gel from the side, if I look at a real image, so this is not from a top, this is from the side of it, you can see I took this video in the lab. This is a time lapse of the dyes moving through the gel. So we add 2 dyes inside of our mixture with the DNA. One of the dyes will move much faster than the other, and it helps to visualize approximately where our DNA is traveling through the gel. So you can see after that amount of time, we have our DNA samples are approximately in this region right here. So they started up here in this little well, and then they moved down and separated. So let's do an example here. We talked about our 4 samples that we digested, 1, 2, 3, 4. We're gonna place our 4 samples in these wells right here. This is my little pipette that I'm adding. And then I'm going to add in, right here, I'm gonna add in a ladder. And this ladder is gonna specifically point out just some landmarks of where the DNA sizes may be. So here's our results. Let's try to analyze them. So first off, let's check. Our ladder looks good. And this will help us identify what's going on. So let's look at our first sample, which is in the second well. Okay, so I'm gonna line them up with the ladder, and you can see, okay, it matches pretty good with 1,000 base pairs. With our samples, just to refresh everyone's memory, we had a 150 base pair insert in a 1,000 vector. So for sample 1, this is probably just vector. So I don't think sample 1 actually has our DNA, our insert in there, which is unfortunate. Okay, so let's look at sample number 2, which is the third well. Well, this is something that's actually can be common depending on what you're doing, but this is what we call a smear. And this is when we get a wide range of sort of gradient of DNA that's been passing through, and this is probably either degraded DNA or just straight up contamination of DNA where it may be either too much was loaded in the well, or we might have DNA of various sizes that are degrading inside of the gel. So this, I will say, is, we'll just say contamination. So either way, we're not getting what we're supposed to get. Okay, let's look at number 3. We can see we have another band, what we call them, and if we trace the band along, we can see, oh, it's a little higher than the 1,000. Okay, that's interesting. This is probably our vector and our insert together, but the digestion did not work, so it is undigested. So maybe the insert might be wrong, we're not really sure, maybe the digestion didn't work. So there may be multiple reasons for that, but that's mostly that's the most likely reason of what's going on is we have our vector here, maybe it has the insert inside of it, and it wasn't cut properly. Okay, so that might need some more, some more tests to know what's going on with that one. Let's look at number 4, our 4th sample. Hopefully this has it. Well, okay, let's look. So we have this one bright band here. Okay, look, matches up with the 1,000. That's great. I'll go back to red I was using before. We have this band down here. Whoa, it's slightly above 100. Let me try to draw that a little better. Here we go. So this one has what looks like our vector plus the insert, and it was successfully digested. That's great. You may be able to notice we have a very faint band right up here that matches exactly with the band next to it. That is probably our vector plus insert that didn't digest. So depending on your digestion time, depending on what you're doing, you might actually, not all of the vectors and inserts will digest completely. It could depend on the efficiency of the buffer you have or the endonucleases you used. But it looks like we have a little bit here that was undigested, but I'm not worried. I would say that sample 4 looks to be good. Sample 4 is best. looks like we successfully inserted our DNA insert into the vector, and then we were able to check that by digesting it and running it through gel electrophoresis. So I hope this helped everyone today. I hope that you can move forward knowing what gel electrophoresis is. All the best. Bye.